I'm Mariana Argenziano. I'm the Associate Director of Manufacturing at Ncardia, and my main role is to develop cell lines and cell differentiation into different cell types from humans and non-humans as well, like human primates and other species, with the application of using this in drug discovery, cardiotoxicity, and safety assays. So there's a growing consensus of that these animals that are highly intelligent should not be used unless this is the latest resort to really get the data that is needed. Of course, we know that shifting away from live animals has been discussed for a very long time, but now we're really making those steps, and I expect that the steps are going to become bigger and bigger as we move away from also other animals. The biggest concern of using NHPs, of course, is that there might be low reproducibility with in vivo systems, the regulatory issues, the cost is very high. So using iPSC-derived cardiomyocytes or iPSC NHPs from NHPs checks all of the boxes by saying we know that they're ethically approved because they're just coming from blood of the animals. So these animals are not being hurt. These animals are not being really suffering to obtain the cells. There's very high reproducibility in producing these cells because we can produce them in large batches, and even within batches, there's still a very high consistency. And of course, the costs are much lower. In the buying a vial of cardiomyocytes is also cheaper than maintaining or housing non-human primates. Well, our platform really addresses what the FDA is asking for. It's really easy to integrate these cells. We really provide the cells ready to use. We provide the cells with the medium, with the protocols. We have protocols for different applications, including electrophysiology, multi-electrode arrays, of course, high-content imaging. And not only we can provide this support online, but we can also support the clients personally and technically in how to develop assays that might not be something that they're used to. We can help with videos, with calls, and that makes it much easier for them to really integrate this, mostly for people or researchers who are not very used to working with in vitro models. It gives the researchers not only the tools to replace their animal models, but also to make sure that they can translate the animal models into an in vitro model, but then also using in vitro human models, they can translate back into the human. So it gives that extrapolation or connection in between the species to make sure that they have the consistency, that they have the biological relevance. But these cells also that we produce at Ncardia, and many people can speak for our human cardiomyocytes. They are very predictive. They are high fidelity, also for cardiotoxicity and safety pharmacology. So we do have very good quality cells that are very consistent, and they can streamline all of these studies as well.